Good morning children, I am Isis. Now I am going to teach you the new chapter, Molecular Basis of Inheritance. As you know, inheritance is the transmission of characters from the parents to generation, next generation. These characteristics are, or these characters are transmitting through a genetic material. So in most of the living things, the genetic material is DNA. So DNA is the genetic material in most of the living things, that is deoxyribonucleic acid. It is a kind of nucleic acid. In living things, there are two types of nucleic acid. They are DNA and RNA. These are the two nucleic acid present in living things. Out of these two nucleic acid, DNA acts as the genetic material in most of the living things. DNA is the deoxyribonucleic acid and RNA ribonucleic acid. So this is the genetic material in most living things. Okay, I'll show you the structure of DNA. So this is the structure of DNA, deoxyribonucleic acid. This is double helix structure. And RNA, it is the genetic material in some viruses only. In some viruses only, the RNA acts as a genetic material. And RNA normally acts as an adapter, messenger, and structural and catalytic molecule. Catalytic molecule in the living things. So these are the major functions of RNA. Okay. So this is about the nucleic acid. And next we can see the DNA in detail. As I told you, DNA is the genetic material, deoxyribonucleic acid. And the DNA is a long polymer of ribonucleic acid, deoxyribonucleic acid. It is a long polymer of or chain of deoxyribonucleotides. It's a long polymer of or long chain of deoxyribonucleotides. Actually, it is made up of two polynucleotides. It is made up of two polynucleotides. And these two nucleotides, two polynucleotides are complementary to each other. I'll show you the structure. So here, this is one polynucleotide and this is another polynucleotide. Such two polynucleotides link together. And these two polynucleotides are complementary to each other. That I will teach you later how it becomes complementary. Now we can see the length of the DNA. So the length of the DNA can be defined as the number of nucleotides. So it is determined by number of nucleotides or number of base pairs. Number of base pairs. BP, base pairs. Base pairs means a pair of or complementary strands of DNA, a pair of nucleotide. I will show you a pair of nucleotide. So here these are the base pairs. So based on the number of base pairs we can define the length of the DNA. So these two complementary strands are called base pairs. The complementary strands are called base pairs. So is it clear about the base pairs? Base pairs means two complementary strands of DNA are called base pairs. Based on the number of base pairs or based on the nucleotides, number of nucleotides, we can calculate the length of the DNA. And the length of the DNA is the characteristics of every organism. For example, in case of human beings, the haploid cell of human beings consists of, not diploid, its haploid cell consists of 3.3 into 10 raised to 9 base pairs. Okay. In case of E. coli, Escherichia coli bacteria, it consists of, the length of the DNA is 4.6 into 10 raised to 6 base pair. So, the length of the DNA is the characteristics of every organism. So, it can be defined by the number of nucleotides present in the DNA or number of base pairs present in the DNA. Is it clear about the length of the DNA? Next we can see structure of DNA. See, the structure of DNA, as I told you, the DNA is a double helix structure. It is made up of two polynucleotide chain. Made of two polynucleotide chains. So, the polynucleotide chain is a condensation, is formed by the condensation of nucleotides. So, the condensation of nucleotides is called a polynucleotides. How does nucleotides form? We can see. Before that, a single nucleotide consists of three components. So, several nucleotides together form polynucleotide. Such two polynucleotides form a DNA strand. Is it clear? And the nucleotide consists of three components. The three components are nitrogen base, pentose sugar, and phosphate group. These are the four components of a single nucleotide. Such several nucleotides, phosphate group. These are the three components of a single nucleotide, and such several nucleotides together form a polynucleotide. Such two polynucleotides together form the DNA strand. Let's see the nucleotides components in detail. For that, we can see the nucleotides in detail. Nucleotides components. The nucleotides consist of three components: nitrogen base, then pentose sugar, then the third one, phosphate group. These are the three components of the nucleotide, a single nucleotide. This nitrogen base are of two types. There are two types of nitrogen base in the DNA, that is purines and pyrimidines. Purines and pyrimidines. The purines and pyrimidines come opposite to each other. Okay, so the purines always bind with the pyrimidines. So which are the purines we can see? There are two purines. They are adenine and guanine. Which are the purines? Adenine and guanine. And which are the pyrimidines? Cytosine and thymine. These are the two pyrimids. Here, in case of RNA, this thymine is replaced by uracil. Uracil in RNA. or in the nitrogen base, the thymine is absent. Instead of thymine, uracil is present in RNA. Is it clear? Now we can see which is the pentose sugar. The pentose sugar is the deoxyribose sugar. Deoxyribose sugar or ribose sugar. We can say the ribose sugar, deoxyribose sugar. And phosphate group is the phosphoric acid. Phosphoric acid. Okay, so these are the components of a nucleotide. And here this pentose sugar and phosphate group form the backbone of the double strand of DNA. And these nitrogen bases are projects in that backbone. From that backbone there is a projection, that is nitrogen bases. Here one more thing I want to tell you. This adenine always base pair with the thymine through 2 hydrogen bonds. So the adenine or this purine is always base pair with this pyrimidine. So adenine always base pair with the thymine through 2 hydrogen bond. If it is RNA, this adenine is base pair with the uracil because thymine is absent there. That is applicable in case of RNA. Okay, but in DNA, the adenine is always base pair with the thymine through 2 hydrogen bond. And this guanine always base pair with the cytosine through 3 hydrogen bonds. Is it clear? So the guanine always base pair with the cytosine through 3 hydrogen bonds. Or otherwise we can say the thymine is always base pair with the adenine and the cytosine is always base pair with the guanine. Is it clear? So we can say the purine is always base pair with the pyrimidine, or we can say purine comes opposite to the pyramid. I will show you the picture. Okay, before that I want to rub it. Okay, you can see. So this is a double helix structure here. So this backbone, this is a single, this blue color is one polynucleotide and this red color is another polynucleotide. Such 2 polynucleotides form this double strand DNA, and this blue color polynucleotide, this color is given for distinguish between 2, and actually it is not blue and red in color. So this blue color polynucleotide here, this area is the backbone which consists of pentose sugar, or we can say sugar and phosphate group. Sugar and phosphate group, because it becomes the backbone of this. And here also this is the backbone of next polynucleotide. And this backbone is formed of sugar and phosphate group. And here also this blue color, this backbone is formed of sugar and phosphate group. Okay, and there is a projection from this polynucleotide, and this is called a nitrogen base. Here we can see nitrogen base. So the first we can see guanine base pair with the cytosine, adenine base pair with the thymine. Again GCTA like that. G always, guanine always base pair with the cytosine. Adenine always base pair with the thymine. Here in turn thymine always base pair with the adenine. Like that you can see the base pairing. Is it clear about base pair? Okay. In the next class we can see how this polynucleotide form. Hope you understood everything about the structure of DNA. That's all for today.